Hi guys and welcome. I'm hoping you are all energized and ready for this revision session. We're looking at a topic known as meiosis. And you know by now that meiosis is found in paper 1. This is where you do meiosis. What do you need to know in this section? In other words, what is examinable? Let's go through that. First of all, you must be able to define meiosis. What is meiosis? State where meiosis takes place in plants and in animals. You need to describe the changes to the chromosomes during meiosis 1 and 2. In other words, you need to follow those chromosomes from the beginning of meiosis 1 right up to the end of meiosis 2. Fortunately for you, you do not need to memorize the names of the phases, but you need to know what happens to the chromosomes during that procedure. You need to then explain why meiosis is important, and then you look at ideas of the consequences of abnormal meiosis. In other words, abnormal, when meiosis goes wrong, what can happen? And we focus on two in our curriculum, that is Down syndrome and polyploidy. We then move on to the advantages of polyploidy to agriculture, in other words, to the farming industry in this country or anywhere in the world. Lastly, we go back to your grade 10 and 11 work, where you would then compare meiosis, which you're doing this year, with mitosis, where you have done in previous years. So this is the crux of what is. Our questions may not cover all of this, but this is what you need to know. These are the things that you need to know. Let's start with the questions. And the first question taken from November 2012, paper 1, question 1.1.6. And that's our first question. The diagrams below represent 6 different phases of meiosis taking place in a cell with 4 chromosomes, all important information. So they give you the diagrams. There are 6 diagrams that are given to you. They want to know the correct sequence of the different phases in which the above mentioned division takes place is. And these are multiple choice questions. So the answers are 1, 2, 3, 4, 5, 6, and it goes on B, C, D. Now let us look at these diagrams. Let's take a moment, pause, and look at these diagrams. Our knowledge on meiosis says that initially, and again, you're not going to need to know the name, but we would say prophase 1. You don't need to know that name. But during that phase, we can see the chromosomes lying next to each other. So keep that in mind. And at the end of meiosis, right at the end of meiosis, we would have 4 cells that have formed. Okay? So that's something we need to think about. All right, let's look at the diagrams. Now, let's look at the first diagram. We can see clearly here somewhere there's about 4 cells that are forming there. Here we got 2 cells and the chromosomes are in the middle. But they are single, 1 each. Here we see 2 sets of chromosomes that are lying next to each other. Here we're seeing something happening with the chromosomes on either side of the midline there. Here we're seeing 2 whole chromosomes moving apart, away from each other. Here we see 2 chromatids. Remember now what's happening here. There are single strands here. Single strands. They're not double strands like here. Here you're getting this situation. They are double stranded, so it's a chromosome. Here we have a single strand, and that is a chromatid. Now, number one thing you must understand, when the chromosomes are together somehow, this will be meiosis 1. And when the chromosomes are single, it will be meiosis 2. So let's look at that now. So this one here will be meiosis 1. This will be meiosis 1. This will be meiosis 2. This one here, we have a question mark because we can't see much happening there. This one here would be meiosis 2 because they are single. And this one here, you can see clearly there's about 4 cells forming there. 1, 2, 3, 4. So this will be meiosis 2. And as I said, meiosis ends when there are 4 cells that are forming. And in this diagram, we're seeing that 4 cells are starting to form at least. So this should be our last number. And remember I said in the start, when we look at meiosis, the beginning we must be able to see the chromosomes lying next to each other. So this number 3 should be more or less our first number. Why? Because the chromosomes are lying next to each other. So let's look at the multiple choice now. And we said that number 3 must be first and number 1 must be last, right? So we look at A. A is saying 1 first. Can't be, right? So we scratch A out. B is saying 6. 6 definitely can't be the starting point. It's in the middle of everything. Out. The next one says 3 is the starting point, and we're saying that's the one, so we just do that first. The next one, but now, wow. There's 2 that say 3 are first. What did I tell you? Which is the last one? The last one is 1. So go to the answers again. There's 1 there, 3 there. So your correct answer, the most correct answer here is C. Can you see, just by going through that, even if you didn't know the other sequence, you can get away. But it's not always the case that in that line. Remember that. But if you can be sure of the first one and the last one, and if you find it like that, but sometimes they could have even put 1 here and then mix 5 and 6 or something else in between. Then you have a problem there as well. So be careful. But the point I'm making here is the elimination process. We move on to the longer questions, adapted again from 2012 paper 1, question 2.1 in the same paper. The diagram below shows crossing over in a pair of homologous chromosomes. Homologous chromosomes is a pair of chromosomes that are lying next to each other. Notice there are 4 strands, 1, 2, 3, 4. 2 strands make up a chromosome. A chromosome is made up of 2 strands. There you go. That's a chromosome. So when they're together, according to their characteristics that they're coding for, then we say they are homologous chromosomes. And that's what is showing there. But also at the same time in this diagram, you can see that something is happening at X. And there is pointing to some structure Z. Identify the point X and part Z. Part Z, that's a part, and point X. That's a simple question, and the answer is X is the chiasma, there, the chiasma, and Z is the centromere. Chiasma, guys, is the part where the chromosomes cross over. That point where they touch. There, this area here. So in other words, the homologous chromosomes are going over each other like that. That point where they are touching is known as the chiasma, or in plural, chiasmata. Okay, that one was chiasma. Give one observable reason why the chromosomes above are regarded as homologous. Why are they called homologous? And they want to know, not from your knowledge, but from what you can see there, evidence from the diagram. And that would be that the chromosomes are of the same structure, size, shape, or length. As you can see, look at them, all right? That's one. And this will come from your karyogram knowledge as well. When you are matching chromosomes, homologous chromosomes with each other, you will match those chromosomes that have the similar shape and size with each other. And we can see also that important process of crossing over that's taking place. The next question says, give one reason why crossing over is important. In other words, you've identified the chiasma, we've spoken about crossing over. Now they want to know, why is this crossing over important? And this answer will not ever change. Whichever way it's asked, whether they show you the crossing over and they say, explain the significance of that process, or whatever way, the answer will not change. The answer will always be that it allows for recombination, gametes, and this increases genetic variation. If you want to put it in its simplest form, we will say crossing over. Remember, you must try and express this in your own words without losing the essence of the answer. So we would say crossing over allows for the exchange of genetic material, and this brings about variation. What we're actually saying, guys, very simply, that when this crossing over takes place, at this point, what will happen? Let's say, for example, let's do some interesting things here. Let's make this line here purple. So now what would happen? At the end of crossing over, that part will remain the same. That part will remain the same. Then you'll have that and a little bit of that. And what's going to happen, if I can manage, that's what you're going to get. So originally, the entire chromosome was green. Okay? There, that one, the left hand, A and B. After crossing over, this is what it's going to look like. So that's what it means. When you say exchange of genetic material, means the genetic material from this chromosome was pushed onto this one. And if we had to draw the other one, this line would have been going like that, stop there. Okay, now that one was green, so we're going to leave it green. Remember, it's not supposed to be, but we're going to leave it green now because we've drawn it that way. And you're going to get that. So in other words, after crossing over, these chromosomes are not the same anymore. They have changed. Why? Because some material from there comes here and some comes here. So now, this is the beginning of the most important significance of meiosis, and that is that the resultant cells would be different from the parent cells. And that's what we mean there. Name one other process occurring during meiosis that has the same importance as crossing over. Now, what importance is this? This importance is related to the fact that it brings about variety. So what other process takes place in meiosis to bring about variety? And that would definitely have to be the random assortment of chromosomes or independent assortment of chromosomes during metaphase. You don't have to say metaphase, by the way. Now, I'm going to extend this page a bit just to explain what we mean by this random assortment. If this is a cell, and the chromosomes, and now I have to be a little creative here. And these were the chromosomes. Random assortment means that there is no fixed way in which these chromosomes here will arrange at the equator. This is one way in which they could have arranged themselves. They could have also arranged themselves undo. Now you see here in this diagram, it's the same chromosome, same homologous. These are homologous chromosomes, these are homologous chromosomes, but they have swapped places. Now, it means that the cells on that side are going to get these guys, and the cells on this side will get these guys. In the same way, we could even move this whole set there and that whole set this way. So there's different ways in which these chromosomes can arrange there. And the word random indicates that there's no fixed way in which it's going to be there. There's no guarantee, you know. That's so much like the lotto. The lotto is a random thing. When those balls come out, it is totally random. If it was not random, if it was fixed, you and I would not be engaging on this platform now. We would rather be just sitting and predicting the next lotto win, so you and I can be sitting there, chilling out in the Bahamas somewhere and having a yacht and everything else, we don't have to worry because we can predict what's happened. But this means that the degree of predictability is close to zero, and therefore there's no guarantee which sperm or which egg will get which resultant chromosome. This also brings about a degree of variety. Good. Now the question says, if a mouse egg cell contains 20 chromosomes, a mouse egg cell is a gamete, and if it has 20 chromosomes, how many chromosomes will there be in its skin? Now, what knowledge is being tested here? Knowledge on meiosis and the result of meiosis. Remember, meiosis takes place for the production of gametes. That means, in humans, to give you an example, spermatogenesis, oogenesis, the formation of sperm, formation of egg, is a result of meiosis. When that last sperm or the last form of the egg is formed, is because of the steps of meiosis that has brought about the changes in that way. And you need to know the story about diploid and haploid. Meiosis takes place in diploid cells to give rise to haploid cells. Haploid means half the chromosome number. So, if the egg cell had 20 chromosomes, then the skin cell, which is a normal cell, it is not a gamete, it's a normal body cell, should have, I hear you, should have double the chromosome number. So your answer would be 40 or 20 pairs. I know you come from the OBE generation, but 20 pairs is the same as 40. That means 20 times 2 equals 40. Moving on to question 3. Notice that the diagrams that are here are similar to diagrams we saw in that multiple choice questions. That shows you how we can use diagrams for multiple choice questions, and we can take the same diagram and reorganize the diagram and ask it in section B. Study the diagram below representing various phases of meiosis in an organism. Diagram A, B, and C. Notice this diagram, the chromosomes complete. Here, single, 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 single. Here, chromatids. Those are your clues. So what, you see those things, that must tell you a story. Which diagram, A, B, or C, represents meiosis 1? Meiosis 1, the chromosomes are together or they are whole. So the only answer there could be diagram A. Suggest why the chromosomes in diagram B will be genetically different from that of the parent cells at the beginning of meiosis. Why would these chromosomes here be different from the original parent? We discussed the whole question on that earlier, and that would be because crossing over has taken place. There was exchange of genetic material, and there was random assortment of chromosomes as well. But the most important part would be up to there especially. How many chromosomes will each daughter cell have at the end of cell division? We look at this one here. How many are there? 1, 2, 3, 4. So if there were 4 in the original cell, then yes, there should be only 2 chromosomes in the end, because meiosis converts the diploid number to a haploid number. Give 2 reasons why this type of cell division is important. Now, remember, some of these questions are talking to our bullets when we started. The previous question on crossing over dealt with the significance, the importance of meiosis. This question is going further with that importance as well. And what are those importances? One is, and that's what we were talking about earlier, it increases genetic variation. In other words, it brings about differences in organisms. That's why you, your brother, your sister, whoever else in your family, you do not all look the same. Imagine a world where we all looked exactly the same. How difficult it would be to solve crime, for example. Okay? It reduces the number of chromosomes by half. Therefore, when that happens, it maintains a constant chromosome number. What do we mean by that? Because the gametes have half, half plus half gives you a whole again. In other words, let's take humans. In a sperm, there's 23 chromosomes. In an egg, there's 23 chromosomes. So when those two fuse again, then the organism that develops from there will have 46. If that was not the case, then the organism that developed would have 46 plus 46 is 92, and the next generation would have 92 plus 92, 184, and so on and so on. This way, because the chromosome number is reduced by half, when the two fuse again, we get the normal chromosome number forming there. It also results in the formation of gametes. It's linked to that one there. All of this is with that. It also allows for alternation of generation, mainly in plants. And you have studied in reproduction paper 2 the life cycle of the moss, the life cycle of the angiosperm. You learned about two different life cycles in them, the gametophyte generation and the sporophyte generation. Meiosis makes it possible because the gametophyte generation is haploid, N, and the sporophyte generation is diploid, 2N. So because of meiosis, the diploid can be converted to haploid, and in that way keep the chromosome number the same all the time. With that, guys, I'm hoping you have whet your appetite for more to come. So take a small break, get the best nourishment you can get in this time, come back and let's kill this section off. Hi, I'm hoping you nourish yourself properly and especially drink lots of liquid, guys. Drink lots of liquid all the time. Liquid and water, not just any liquid. Water is the healthiest option. We're still busy with meiosis and we're looking at some other options. Notice, guys, as we're going through, we're seeing different diagrams and we're seeing different questions. So in this 1 hour session that you're doing revision, we are looking at a variety of ways in which this section can be tested. This is the whole idea. Let's move on. Study the diagram below showing a process during meiosis. Now they're just talking of a process during meiosis. And there you have A, B, C, D. Notice it's labeled clockwise. And they're saying label structures A, B, C, D. Okay, I'm going to do it on the diagram and then we will open it. A would be easy. That would be centromere. By the way, centromere is that structure which holds the two chromatids together. B is pointing to this structure here. That would be a chromatid. The most correct answer would be chromatid. However, in the exams, they may also accept chromosome. But in this exam, they wouldn't have accepted chromosome. Why? Because of this label here. Can you see this label C? C now has a curly bracket, and that becomes chromosome. So where in a diagram, it's a nice example, where in a diagram they have pointers to different things, then they won't give you the option of giving you either or as an answer. Okay? And D now, look at D. D has a curly bracket with these two guys next to each other. So here you needed to be even more specific and you had to say homologous chromosomes. Now some textbooks call this structure like this bivalence. But we look in our syllabus, we are talking about homologous chromosomes. Let's look at the answers. Okay. Centromere was A, B was a chromatid, chromosome, and D was the homologous chromosomes or bivalent. Which process is illustrated in this diagram? Guys, from the last two questions, this question, remember in an exam, you're not going to get three of the same type like this. This is for revision. So here now it becomes easy because what process is illustrated there? Simple crossing over. During which phase of meiosis does the process named in 4.2 take place? Now, I'm going to change that to during which stage, because you are not necessary, it's not necessary for you to know the names of the phases. So we'll keep it to the stage. During which stage of meiosis does this take place? Obviously our answer will change, and now I'm going to leave the same answer there, but I'm putting that in brackets. Prophase 1. It takes place in the phase known as prophase, but in meiosis 1. So prophase 1, which is important. Crossing over takes place in the stage 1. And that you need to know, the difference between stage 1 and stage 2. If a diagram is showing crossing over or chromosomes in metaphase or in the middle phase lying next to each other, or when they're separating, the whole chromosome is separating, or at the end there are two cells formed and the chromosomes are still whole, that will all be stage 1. Stage 2, the homologous chromosomes are not going to be together at any stage. And when they're separating, the chromatids separate and move to opposite poles. And in the last stage, there will be 4 cells. And in those 4 cells, there will be single strands, just chromatids initially. And after interphase takes place, DNA replication takes place, those chromosomes become whole again. So you need to know that as well. What is the benefit of the process named in question 4.2? We've spoken about already. It ensures that the gametes produced at the end of meiosis are genetically different from each other. In this case, they only gave 1 mark. If the same thing asks for 3 marks, significance, then you'll start by saying about, what did I tell you earlier? That genetic material is exchanged, and this leads to variety. So you're getting your mark for genetic material exchange. Other textbooks talk about there's a shuffling of genetic. It means they're changing from one chromosome to the other. Name and describe. Now what's this question? And guys, many of you get this wrong. Whenever there's an and between two verbs, in this case, you tend to leave out one or the other. So be very careful when that word and is there. There's 2 things you have to do. Name and describe. One other way in which meiosis is able to produce the same benefit. Notice this question is very similar to the one we spoke about earlier. Now here, but you have to describe it as well. So we're naming it as random or independent assortment of chromosomes. And then we say either the chromosomes or the chromatids arrange themselves randomly in no fixed order, independent on either side of the equator during metaphase 1 or 2. This part here, you don't have to mention. Up to there is what's important. So all you're saying is random assortment means that the chromosomes in stage 1 or the chromatids in stage 2, whenever, they arrange themselves in a very random order. This brings about variety. Question 4 says, tabulate 3 differences. Tabulate, verb, how many? 3, what? Differences between meiosis 1 and meiosis 2. And first and foremost, you have to draw a table. That's your 1 mark. You're going to get 1 mark just to draw the table. And then you're going to, headings will be there, meiosis 1, meiosis 2. During meiosis 1, crossing over takes place. Here there's no crossing over. We could even add here, and this is more important, even in meiosis 1, homologous chromosomes lie next to each other. And you can say this in both these things here, they mean the same thing. Not the crossing over the homologous chromosome, because this happens in prophase 1 as well. And in matter the chromosomes align in homologous pairs. In stage 1, the homologous pairs align themselves together at the equator. And in meiosis 2, they align singly on the equator. Meiosis is a reduction division. Meiosis 2, there's no reduction there. In meiosis, whole chromosomes move towards opposite poles. In meiosis 2, chromatids move towards opposite poles. And that's what we were talking about there, prophase 1 and prophase 2, about homologous chromosomes. You needed to give 3, and guys, when they tell you about giving 2 or 1 or 2 or 3 or 4 or 5, make sure that if you're giving more than that, your first 3 in this case must be correct. Because if your first 3 are incorrect and the last 3 are correct and you gave 6, you will still get 0 unless you have got 1 mark for the table. Because the memo would say mark the first 3 only, or mark the first 1 only, etc. When they say list 1, list 2 or whatever, name 2 or whatever, that's what you need to keep in mind. You can't mess around there, because if not, you imagine you do all that effort and you get everything wrong. So prioritize your answer. Question 5, coming from this year's March paper, version 2. Remember, up to March of 2013, there were 2 versions of the paper written. The paper versions are different from each other in the content in paper 1 and paper 2. However, certain sections like DNA, RNA, protein synthesis, meiosis, etc., they are, the content is the same. Reproduction, although reproduction is now in paper 2, in the past it was in paper 1. Evolution was in paper 2, now it's in paper 1. But the content is still relevant. Not all of it, but some of it. You need to be careful. But these questions also for meiosis, protein synthesis, etc., you can go to right up to 2008 and look at those past papers as well. All right, so again, animal cell in a phase of meiosis. Look at the questions we're asking. Again, label A and B. In this case, A, again, the chromatid. And B now is a different label from the past, and that's these things that are pulling the chromosomes apart or holding them in place, and this is known as the, that's it, spindle fiber. Good. State where the phase of meiosis shown above, whether the phase, my eyesight's failing me, whether the phase of meiosis shown in above is part of meiosis 1 or meiosis 2. Again, remember I told you they can ask you that. Now here, where are the chromosomes? The chromosomes are lying at the equator, middle of the cell. And again, I must stress at this point that the middle of the cell doesn't always mean middle from top to bottom there. It can also be, like in this diagram, it's showing you side by side. Here's the spindle fibers forming that way. So therefore, your middle of the cell is there. And they're lying in the middle how? The homologous chromosomes are lying next to each other. And if that's the case, it will be, as I told you earlier, stage 1, meiosis 1. Give one visible reason for your answer, and I've given you that already. Chromosomes are lined at the equator of the cell in homologous pairs. If it was meiosis 2, they would be at the equator singly, alone, one only, per spindle fiber. How many chromosomes are present in the cell above? And I have to stop and pause at this stage. Amazing, amazing, and more amazing. Here's the cell. Now, how many chromosomes can we see in the cell? How difficult can that be? Let's see. 1, 2, 3, 4. Are there any more chromosomes at the back? No, I can't see any at the back. Above this, besides this, nothing. There are only 4 chromosomes. How do we know what a chromosome is? A chromosome is a structure which is made up of 2 chromatids, tied together with a centromere. This makes up a whole chromosome. So we are looking for whole chromosomes here. There's one strand, two strand, centromere. One strand, two strand, centromere. One strand, two strand, centromere. 1, 2, centromere. So it gives us 4. Many, many thousands of learners get this wrong, and I can't understand why. It's a simple question, so our answer is obviously 4. I was scared to actually click on that answer in case my answer was incorrect, and I'm telling you about how your answer is. But that's why I'm getting worried. How come people are getting this thing wrong? Good. How many chromosomes will be present in each cell at the end of meiosis? If there were 4 at the beginning of meiosis, what will there be at the end of meiosis when meiosis' job is to reduce the chromosome number by half? 4 divided by 2 should, I hope, you think I'm right, give us 2. Good. Simple. So at the beginning, there's X amount. In the end, there will be X divided by 2. Whatever the number is, divided by 2. Remember, it's not always 23, because meiosis is not only taking place in humans. That's the other answer we get in this question. How many chromosomes are there in a cell? 46. Why? Because it's diploid. But we didn't ask you in the sperm. We didn't ask you in the egg. We didn't ask you in the skin of humans. We asked you in this diagram here. How many chromosomes can you see here? Okay? Keep that in mind. That's a very, very common mistake. And at the end would be whatever that number is divided by half. 23 is in our bodies. Our sperm and in the females, the egg has 23. That's half of 46, which we have in every other cell in our body. Could the cell represented in the diagram be that of a human? I didn't know that question was coming. I gave you the answer already. No, it can't be, because why? Explain your answer. Good. They're asking you already. Explain your answer to question 5.5. What is the reason for that? And the reason is that there are 4 chromosomes present there. Sorry about that. 4 chromosomes there, whereas in humans it's 23 pairs or 46. This is a diploid cell, and the diploid number in this cell is 4. Whereas in humans, the diploid number is equals to 46 chromosomes. And we don't just have 46 chromosomes. That's why we're stressing this 23 pairs all the time. In all organisms, whatever it is, here also we should say 2 pairs. So stressing. If you say 4, there's nothing wrong with it. But why are we putting this 2 pairs and 23 pairs? So that you understand that it's not just 46 chromosomes. There's 23 chromosomes that you got from your mother, 23 chromosomes from the egg, 23 chromosomes you get from your father, sperm. And that makes your 46. And these chromosomes are specific in the sense of their size and their shape, and more importantly, on what they code for, whether they're coding for your eye color, your size of your nose, your bum, whatever else that they're coding for, they are specific. So a different chromosome in your body codes for these different things. I'm hoping that helps you understand that. We move on to another question, and this question is question 6. So the question says, describe. There you go. The first word is the verb here. The verb, and this verb is addressing you, the learner. Describe. Describe what? Whatever comes after it, if I'm going to tell you, fetch, I can't say bicycle fetch. I'm going to say fetch the bicycle. So the verb is given to you. Whatever comes is what you must do. Fetch the bicycle. So the same way, describe. What must you describe? You must describe the changes that occur to the chromosomes during meiosis 1 and 2, and another verb. Compare meiosis to mitosis. 2 things that you have to do here. One is you've got to describe the changes that occur in the chromosomes during meiosis 1 and 2. Notice they're not asking for the phases and so on. They say, tell us what's happening to the chromosomes. So which part was the first part of the question? This one here. Describe that one. So that becomes number 1, and that becomes number 2. Not that you're going to number it, but preferably, preferably you answer it in that order, because the memo 9 out of 10 will be in that order. So when a person is marking it, it flows with that as well. Makes it easier for the marking. And whatever makes it easier for the marking is actually in your favor in the end. So first we must talk about meiosis 1, and we must explain. Now we're not going to write everything down here. We're just going to talk about the major events that are going to take place, and you're going to elaborate. The idea is to get you the skill. So first of all, in meiosis, what happens is that the chromosomes become visible and the nuclear membrane disintegrates, spindle fiber forms, and homologous, this is important now, homologous chromosomes lie next to each other. And crossing over takes place. You'll take another 2 lines to explain what is meant by crossing over, that what it does and the significance. So now you've told us what happens to the chromosome then. Thereafter, the chromosomes, still in stage 1, arrange themselves at the equator, still in pairs, homologous pairs. So you're telling them they're still lying in pairs. Then the spindle fiber contracts, and if you know your phases, you'll follow me even easier here. The spindle fibers contract, pulling each chromosome of the pair. What do we mean by that? One of the two chromosomes are lying next to each other, one is pulled one way or the other towards opposite poles. I'm hoping you're understanding that this encapsulates the whole of meiosis in one question. So pulling them towards opposite poles. Then the chromosomes arrange at the poles. Remember, because the question spoke about the chromosomes only, there's no need to talk about the other stories. But you can, if you want to make it more complete, you say the nuclear membrane reappears, you say the cell pinches up, and you have now 2 cells. With, and it's important, this last part, 2 cells form with half the chromosome number. And that brings us to the end of meiosis 1. In meiosis 2, now we're going to move on. Meiosis 2. What happens in meiosis 2? We don't really have a prophase 2 as such, but the nuclear membrane will disappear again. Now what will happen? The chromosomes then arrange where? At the equator. But now they arrange singly. They no more in pairs. And then again the spindles contracts, pulling. Now what does it pull? Because they're single, if one is going to pull one chromosome, that means only one is going to go one way. Pulling chromatids apart. And for this to happen, the centromere splits to allow this to happen. At the end, the chromatids now are arranged at the poles for different cells result. So you have given the story. What have you done? You have told a story. What story? The story of the chromosomes in meiosis 1 and in meiosis 2. Is that the end? Not at all. And compare meiosis to mitosis. Now, this second part, believe it or not, is very similar to this question. However, you're writing an essay, so you're not going to put it in a table. Why I say is this very similar to this? Because meiosis 1, meiosis 2, the differences. Meiosis 2, you'll see your textbook says, is very similar to mitosis. So the differences are going to be, the same. What are we going to say? In meiosis, let's go and we write our own story, because we are writing a story. During meiosis, the homologous chromosomes lie next to each other and crossing over takes place. This does not occur in mitosis. The homologous chromosomes need not lie next to each other. Further, the homologous chromosomes in meiosis, the homologous chromosomes lie next to each other at the equator. This does not occur in mitosis. In meiosis 1, you've got to be specific in this difference. In meiosis 1, chromosomes move to opposite poles when the spindle contracts. In mitosis, yes, chromatids. Notice chromosomes, chromatids. And notice we have to say meiosis 1 because in meiosis 2 the same thing that happens in mitosis happens. Okay? And also here, let's be specific here. In meiosis 1, the chromosomes lie next to each other. Even here, you've got to say in meiosis 1. Where is that? During meiosis 1. In all our answers there, we must talk about meiosis 1 because in meiosis 1 that doesn't happen. There's no crossing over. Homologous chromosomes are not lying next to each other. At the equator also in meiosis 2, the homologous chromosomes are not lying next to each other. When the spindle contracts in meiosis 2, it's chromatids, the same like this. So that's why you have to be specific. In mitosis, the chromatids move to opposite poles. Compare mitosis, and so they didn't give you specifics, so you can go to town with this question. You can go to town with this question. We can add to this question, for example, mitosis results in 2 cells. Whereas, it's another linking word you can use is whereas when you're comparing 2 things in an essay. Whereas meiosis results in formation of 4 cells. Mitosis is exact duplication. In other words, duplication, wow, what a bad speller. Duplication. In other words, identical cells form. Whereas in meiosis, variety is introduced. Or you can say the cells that form are different from each other. We can go further. Meiosis results in gametes forming, whereas mitosis are ordinary somatic cells forms. You can go further and talk about meiosis is for the production of gametes and so on, whereas mitosis is there for repair and so on, because the question did not say in terms of the process, it just said compare mitosis to meiosis. So can you see we filled in quite a bit of stories there. Remember, for each fact that you give here, there is 1 mark. You need, normally in an essay, there are 20 marks given. 20 marks are given, of which 17 marks are for the essay and 3 marks is for synthesis. Now, the 3 marks for synthesis does not mean that you have to write a heading, synthesis, and write something under it. These 3 marks, there's a rubric in the marking memo which will guide the marker to allot these 3 marks. I can tell you that they are dealing with the following: is answering the question, is the learner answering the question? Secondly, is the information given relevant? Have you answered the question relevantly, or you're giving a whole lot of story? Whatever you know in the section, just throw it in here, and hopefully in between there will be some bits. And do you follow a logical sequence in your answer? In other words, is your answer now telling a story? The bottom line of logical sequence means your answer must tell a story. It must take me through this process. If you're explaining something, it must take me through step 1, step 2, step 3, step 4. You can't start with step 1, go to 4, come back to 2, go back to 3, go back to 1, go back to 3, go back to 4. You're jumping all over the show. That means the marker must look for your facts throughout that essay. Whereas if they're flowing, and remember the trick I told you, look at what the first part of the question is asking, answer that. Look at the second part, answer that after that. And you follow. Do it properly, and look at when I did the differences. I didn't give you all the stories of mitosis and then meiosis. I dealt with each one. This is what happened in mitosis, this is what happened in meiosis. So you finish that idea, you go to the next idea, and so on and so on. This is also true for your creative writing, whatever, English, Afrikaans, whatever language you're writing. When you're writing creative writing, take a paragraph, finish that idea, go to the next, the next, the next. Don't jump all over the poor show when you are writing your essays in that sense. In life science, the essay is not such a heavy essay that you need an introduction, a conclusion, and a body, and so on. We want the facts. The facts must be relevant, and they must follow in a logical sequence. And then you would get the 3 marks that will give you synthesis, and if you had your 17 facts, you'll get 17 plus 3, 20 out of 20 in an essay. Try to keep your essay, 17 marks, 17 facts, can't take you more than a page. The essay in life science is not more than a page. Actually, all right, if you write a little bit big, maximum should be 1.5 pages. Don't write more than that. If you're writing more than that, you're writing too much. That means you will have many irrelevant facts. And once you have that, you're going to lose the 1 mark at least from the synthesis. So keep that in mind when you're writing your essays. And I'm hoping this last question tied up the section on meiosis to do a revision on the whole procedure. Remember, you also have to know what goes wrong in meiosis and how does this cause Down syndrome and some of the symptoms of Down syndrome, and also what is polyploidy and how does polyploidy help in agriculture. I'm hoping this session helps you to revise, and I'm hoping you take this information, you empower yourself, and you knock the socks off any question on meiosis you may encounter. Thank you.